Hello everyone and welcome to week 2. Today we are going to do an overview of the biochemical processes. There are 8 main biochemical processes that you will need to become familiar with for this course. The first biochemical process that we're going to talk about is phosphorylation and dephosphorylation. So to begin, ION, when that's added to the end of a word, it means action of. So in this case, it's the action of removing or adding a phosphate group. So when it comes down to phosphorylation, it's the action of adding a phosphate group. It's achieved by one enzyme and one enzyme alone, and that's called the kinase enzyme family. So one of the main reactions in the body that uses phosphorylation is the creation of adenosine triphosphate, or ATP. It is formed when we add a phosphate group to ADP. So adenosine diphosphate has 2 phosphates. When we add another phosphate, we get adenosine triphosphate, meaning we have 3 phosphate groups. And this is the picture on the screen, on your slide. You can see a big molecule, and that's ATP. We see that it has 3 phosphate groups, and that's dictated by the P, which indicates a phosphate group. This is the main reaction found in energy metabolism. It's the phosphorylation of ADP to ATP. I will be posting in a separate file on Canvas 2 reactions that you should become familiar with, which are phosphorylation reactions. One is the creatine phosphate reaction, which is an immediate energy creation process to form ATP, as well as ADP combining with a phosphate to form ATP. Both of those reactions are energy producing pathways, so they form ATP. We do not have energy released yet. We need to break down ATP to release the energy for movement, but we'll get to that on further slides. So looking at this slide, we're looking again at the action of adding a phosphate group to another molecule. This is the process known as phosphorylation, and it requires a kinase enzyme. So the big molecule that's highlighted in gray is an entire phosphate group. When we talk about adding a phosphate, we're talking about adding, yes, a phosphate, but also 4 oxygens and 2 hydrogens. So a phosphate can be in multiple forms, but it's typically a phosphate is bound to at least 2 oxygens and some hydrogens. So it's a large molecule. So as you can see in the reaction below, we have an R group, which is just a random molecule with a hydroxyl group or an alcohol that's being added to a large phosphate molecule. And if we go in a reaction to the right, follow the A arrow, we form a new molecule, which has R bound to a large phosphate group, and then we have the removal of water. This is known as phosphorylation. We're adding this large phosphate group to another molecule, and that other molecule is denoted by R. So we're adding the large phosphate group to the R, forming a new molecule that now has a phosphate with it. Now let's talk about the opposite reaction of phosphorylation. This is known as dephosphorylation. It's the action of removing a phosphate group from a molecule. So it's the removal of a phosphate group, and this is achieved through the enzyme phosphatase. So you need a phosphatase enzyme to remove a phosphate. If you don't have the enzyme, the reaction will not happen. So let's look at our equation down below. We have, in this case, we're going in a reaction to the left. So we're now removing the phosphate from the R molecule. So we have our water reactant combines with our R molecule that is bound to a phosphate, which is highlighted in yellow. We move to the left. The arrow is moving to the left. We follow the B arrow. And now we have the phosphate group bound to a hydroxyl group. And then we have our R molecule bound to a hydroxyl group. Okay, so we just have removed the phosphate, which is now bound to a hydrogen. And now we have our R molecule bound to a hydroxyl group. Okay, this is the process known as dephosphorylation. We're removing a phosphate from a molecule. Our next biochemical reaction is hydrogenation and dehydrogenation. So it's the action of adding a hydrogen or the action of removing a hydrogen. So let's start with hydrogenation, which is the action of adding a hydrogen. It's interchangeable with reduction. You may have heard about this in high school chemistry. So hydrogenation and dehydrogenation is analogous to reduction and oxidation. Technically, they are different. One involves hydrogen atoms and the other one electrons, but in reality, it's not really that different. So oxidation and reduction are often performed using full hydrogen atoms. So we can generally use these terms interchangeably, and we will throughout this course. So just remember that hydrogenation also means reduction. And in this case, what we're doing with hydrogenation is it's the addition of hydrogen to a molecule. So in this case, if we're adding hydrogens, we're actually going to break a carbon-carbon double bond. Okay, so that's the key here. We're breaking a carbon-carbon double bond. So reduction refers specifically to the addition of electrons, and in this case, it can also be hydrogen atoms. And with every reaction, we need an enzyme to expedite the process. And in this case, hydrogenation is achieved by a hydrogenase or a reductase enzyme. So let's look at the reaction down below. We have a molecule that has a carbon-carbon double bond on the far left. That's our reactant. We combine a molecule that's R, denoted by R. It doesn't matter what it is, but it has two hydrogens bound to it as H2. When we move in this reaction to the right, so we follow the A arrow, we now have we've added these two hydrogens from the R reactant to the carbon-carbon double bond. So we've broken the carbon-carbon double bond. And I've highlighted in the molecule where those two hydrogens have gone. They've gone to the two carbons, and we've broken that carbon-carbon double bond. And now the last product is just an R, which is a random molecule. Okay, so the key here is that with our reactants, we've added hydrogens to a larger molecule, we've broken the carbon-carbon double bond, and now we have a new molecule that has no double bonds. It's fully saturated with hydrogens, and we get an extra molecule, which would be R. The opposite reaction to hydrogenation is dehydrogenation, which is the action of removing hydrogens from a molecule. This is also known as oxidation, where we're losing electrons or losing hydrogens. So it's the removal of hydrogens, and we're actually forming carbon-carbon double bonds. We're forming double bonds because we're removing hydrogens. So oxidation refers specifically to the removal of electrons. In this case, it can also mean the removal of hydrogen atoms. And this is achieved, again, we need an enzyme to achieve these processes. This is achieved by a dehydrogenase enzyme. So you need to remember that, because whenever you see a dehydrogenase reaction, you need to know automatically that it's a dehydrogenase enzyme involved. So in this case, when we look at a reaction at the bottom, we're moving to the left. So we're following the B arrow. So let's start to the right. We have an R molecule, R plus molecule, and then we have a larger molecule that has two carbons and four different R's, which it doesn't matter what they are, but we have two hydrogens in the bottom, okay, bound to the two carbons. If we move this reaction to the left with a hydrogenase enzyme, we remove the two hydrogens, they bond to the R reactant, and now we have our new molecule that is two carbons, but it has a double bond. So we've removed the hydrogens from the carbon molecule, and we've added those to the R molecule. So it's a dehydrogenation reaction. We've removed hydrogens from the main molecule of the reaction. This is called dehydrogenation. The easiest way to determine whether it's hydrogenation or a dehydrogenation reaction, or what biochemical process it is by counting the carbons and counting the hydrogens. If you see that in the reactant there are more hydrogens and in the product there are less hydrogens, we would say that this is dehydrogenation. We're removing hydrogens. But if we see that we're adding hydrogens to a molecule from going from a reactant to a product, we know it's hydrogenation. Okay, so always count the hydrogen atoms because this will help you determine whether it's hydrogenation or dehydrogenation. Okay, the next set of biochemical processes we're going to talk about is hydration and dehydration. So hydration, let's begin with this, is just like what we do when we drink water. We're adding water to our body. So hydration is the addition of water to a molecule without splitting or cleaving of the molecule. So you're not splitting the molecule when you're adding water. This is important because we're going to see this happen later on in another reaction. Okay, so it's the addition of water without splitting of the molecule. And this is achieved, of course, we need an enzyme for this to expedite the process. Otherwise, it would take way too long and we would die before the reaction would happen. So this is achieved by a hydratase enzyme. So we have our molecule denoted by, on the left, so we're looking at the left side of this reaction right now. We have a carbon-carbon double bond, and we have 4 R groups. This is our molecule. We are now adding water to that molecule, and we're moving this reaction to the right following the A arrow. And now you can see one molecule on the product side with water in it now. So water is 2 hydrogens and an oxygen. So we formed a carbon-hydrogen bond and a carbon hydroxyl group bond, or an alcohol bond. So essentially hydration is just adding water to a molecule without breaking or splitting the molecule. The opposite reaction to hydration is dehydration, which is the action of removing water from a molecule without joining the 2 molecules. This is achieved, again, you need a different enzyme, a dehydratase enzyme. So again, this reaction, we're moving to the left. So we're following the B arrow to the left. So let's look at the reactant in this case is a carbon-carbon atom that is bound to a hydrogen and the other carbon is bound to a hydroxyl group. We go through the process of B to the left, and we're removing water from that molecule, and now we have a carbon-carbon double bond. Now we have water as a product. Okay, so it's essentially the opposite of hydration. You're moving this reaction to the left through the B reaction, and you're removing water from a molecule. You're dehydrating it. So you can determine which reaction this is by looking at where water is. If water is a reactant and you're going from one molecule to one molecule with the addition of water, you know it's hydration. If the water is on the product side, you know that you're removing the water, and therefore it's a dehydration reaction. The key here is that you have one molecule. You're not splitting the molecules. You're going from one molecule to one molecule with the addition or removal of water. The fourth set of reactions is hydrolysis and condensation. So let's begin by hydrolysis or hydrolysis. Lysis means splitting of, and then hydro means water. So the word itself means splitting of a molecule with the addition of water. So we're adding water to a molecule with the cleavage of the molecule, with the splitting of the molecule. So remember, this is different than hydration, because hydration, yes, we had the addition of water, but we didn't split or cleave the molecule. In hydrolysis, lysis means you're splitting. You're splitting the molecule as a result of adding water. So let's look at and this is achieved through a hydrolase enzyme. So now let's look at the reaction below. And again, this is a reaction, so we're moving from the left to the right. So we have a molecule that has a carbon-carbon bond, and we're adding water. And as we move to the right through this reaction, we see that now on the product side, we have 2 new molecules. One that has a new hydrogen bond, and the other one that has the carbon hydroxyl bond. We have just split the molecule. So we've gone from a large molecule to 2 smaller molecules because of the addition of water. This is called hydrolysis or hydrolysis. So essentially, we're going from one molecule to 2 molecules when we add water. This is key. This is a very important point that you will be able to identify the difference between hydration and hydrolysis by the number of molecules that are produced. Now let's look at the opposite reaction of hydrolysis, and this is known as condensation. So now we're moving from the right to the left. Okay, we're removing water from two molecules and we're joining those two molecules together. Okay, so we're going from two molecules to one larger molecule with the removal of water. So let's start on the right side of this reaction. So we have our two molecules, one with a hydrogen chain and the other one with a hydroxyl chain to the carbon. If they move leftward through the B reaction, we see that those two molecules are joined together into a carbon-carbon atom molecule and with the removal of water. So we've seen this before, and this is in the formation of maltose, as well as sucrose, as well as all the disaccharides, where they've formed a glycosidic bond with the removal of water. So this is a condensation reaction. So glucose plus glucose is maltose. That's a condensation reaction where we're removing water, and they formed, in that case, a glycosidic bond. Here, what we see is that we go from two molecules to one larger molecule with the removal of water. That's called condensation. It is different than dehydration because you are splitting, well, you're not splitting the molecules, you're adding the molecules together to form one larger molecule. You're going from two molecules to one larger molecule. The last one I want to talk about is dissociation, and this is the process of splitting of larger into smaller molecules. So as you can see in this reaction that's highlighted in gray, if we move to the right, if the reaction goes to the right, we see that it is reversible because there's multiple arrows. But in this case, we're looking to the right. We're going from a larger molecule that has an R chain with a carboxylic acid moving to the right, and we're splitting it into two smaller molecules. This is called dissociation. So when looking at this reaction, there's two things that I want you to draw your attention to. I want you to be familiar, firstly, with the functional group in the reactant molecule. So I've pointed to that in black. I've highlighted the functional group. And you should be able to identify that because you've seen it before when we talked about carbohydrates. Okay, so have a think about that for a minute. What is the functional group in the reactant molecule that I've highlighted in black? By the end of this class, you should be able to identify that no matter what in any molecule. The second question to challenge your learning is what is the functional group in the product molecule, which I've highlighted in red? What is the carbon double bound to an oxygen, single bound to an oxygen? What is that functional group? Have a think. Now let's go over the answers. In question number 1, what is the functional group in the reactant molecule? A carbon double bound to an oxygen with a single bond to a hydroxyl group or an alcohol is known as a carboxylic acid. So whenever you see that in a molecule, you know that that's a carboxylic acid chain that's on there. Okay, that's the functional group. Now let's look at question 2. What is the functional group in the product molecule, which is a carbon double bound to an oxygen, single bound to an oxygen? This is known as a ester. So that's an ester. Whenever you see a carbon double bound to an oxygen, single bound to an oxygen, this is known as an ester. So you need to be familiar with those functional groups, and you may need to refresh your memory on functional groups from high school chemistry. So now a couple more questions for you to challenge your learning. So this is a typical test question. Identify 1, the type of reaction occurring, and 2, the functional groups present in the reactant and product molecules in both of the following examples. So let's look at the first example, which is the top gray highlighted reaction. So let's move, again, it's moving from the left to the right, this reaction. So we have two molecules undergoing a reaction. Now we have one single molecule with water removed. Have a think about that. The second reaction, we have one molecule going to one molecule. based on the 8 biochemical processes we just went over in class, you should be able to answer both of those questions. Okay, the type of reaction occurring and the functional groups present in the reactant and product molecules. Now let's talk through the answers. Let's go to the first example at the top. So we're moving, again, this reaction is going from the left to the right. So remember, on the left is the reactants, on the right would be the products. So on the left, we have 2 molecules, which are going to 1 product plus water. So based on what we just learned about the different biochemical processes, I would suggest that this is a condensation reaction. So we're going from 2 molecules. They're cleaving together to form 1 molecule with the removal of water. So this is known as a condensation reaction. Remember, when we talked about carbohydrates and disaccharides, we had glucose combining with glucose to form maltose. That would be a condensation reaction because water was removed and you're going from 2 molecules to 1. Okay, so what are the functional groups found in the top reactants? So we have alcohol in the first molecule, then we have a carboxylic acid, which is a carbon, double bound to an oxygen, single bound to an alcohol. And then in the product side, we have an ester, a carbon, double bond to an oxygen, single bond to an oxygen, and we have our water molecule. Okay, so that's question 1. It's a condensation reaction moving from the left to the right. In the opposite direction would be a hydrolysis reaction. The bottom example, again, we're going from 1 reactant to 1 product. So the simplest way to do this is to count the carbons. So if we look at the reactant on the left-hand side, we have 2 R's, we have 1 carbon, we have 2 hydrogens, and 1 oxygen. When we move to the product, we have 2 R's, that hasn't changed. We have 1 carbon, no hydrogens, and 1 oxygen. So what is happening to the hydrogens? Well, from going from the reactant from the left to the right, we're removing 2 hydrogens. So what reaction is that where we're removing hydrogens? We know this to be a dehydrogenation reaction, which would require a dehydrogenase enzyme. Okay, so we're removing hydrogens. The easiest way to determine the reaction is to count the atoms in each of the reactant and product and see what is missing. And in this case, the hydrogens are missing. So we know they've been removed from the reactant. In this case, we know that is dehydrogenation. So what are the functional groups found in the reactant? The reactant has an alcohol, or a hydroxyl group. That would be the functional group. Okay. So that would be a great example of something that you would see on a test, is identify what the reaction that is occurring, in which direction, and the functional groups of the reactant and the product. Now what I would like you to do is work through these 4 reactions and determine in each direction what the reaction is and what the reactants and product functional groups are. Okay, so this is the slide that doesn't have the answers. I want you to challenge yourself with this. Remember, whenever you see a P group, it's a phosphate. So you know it's the addition or removal of a phosphate group. So the first one, you should know that it's a phosphorylation or a dephosphorylation reaction. You just have to determine in which direction is which reaction. Okay, so I want you to work through these 4, and then on the next slide, you're going to see the answers. So use this slide as a challenge to challenge your knowledge and your ability to identify reactions based on what you've learned in class so far. And then use the next slide to mark your outcome. How did you do? And what do you need to improve on? As mentioned, this slide goes over the answers to the previous slide's biochemical reactions, whereby I wanted you to identify the biochemical process and the functional groups found in the reactant and the products. After that, identify what you need to improve on. So what you're struggling with. If you have questions, feel free to ask me via email or in our Google Meet extra help sessions on Mondays. Thanks very much. That's it for today.